Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. As I bet to Dr. Zarina, Dr. Christopher, and Dr. Chibuong for willing to actually participate in my presentations, whereby for today I am presenting the topic of my research on the study of polyphenolic compound concentration and antioxidant activity in different apple cultivars from Kundasang. So the apple cultivars are uh, Alice, Brenda, Katie, Dorian, and Emma. So these are my flow charts. I'll be starting with the introduction, then going to the literature review, justification, objectives of my research, materials and methodology I'm using, result discussion for the result, and as well as the conclusion for the last part. Alright, so for the introduction, apple, or the scientific name Malus domestica, is one of the most important and wide variety fruit sources around the world. And why Apple has become this famous and has become always been minded by people, it's not only because of the taste and the structures of the apple, which is so good, but it has been also always been associated with the reduced risk of degenerative diseases such as cancer and cardiovascular disease, such as stated by Wolf 2008, which all of these are contributing to the causes of death in developed countries. And this association is actually attributed to the polyphenolic antioxidants that is contained in apples, which can protect the human body against oxidative stress by scavenging oxygen-free radicals. So free radicals is actually natural occurring in the human body. We humans, we produce these free radicals, and this free radical is actually back to our health by these free radicals are going to like break the bond that are in our human body. So this is why the importance of this polyphenolic is seen to be effectively scavenging the activity of these free radicals inside our body. So many studies have shown that the concentration of polyphenolic compounds such as flavonols and anthocyanins in apple different cultivars. So this is why I'm using different cultivars in the experiment. It also differs between maturity stages, environmental conditions, as well as the part of the fruits. So another factor that I'm going to be studying in my research is the peel part and the flesh part. So according to many authors, actually the content of the compound and the antioxidant activity is particularly high in the peel of apples than the whole fruit apple in the flesh. So that's why I'm going to be proving that actually the concentration of the polyphenolic compounds and the antioxidant activity in the peel is higher rather than in the flesh. So polyphenolic compounds are usually referred to as a diverse group of naturally occurring compounds that contain multiple phenolic functionalities. But this natural occurring is always happen in higher plants, and they also have the medicinal and industrial value for potentially treat numerous biological activities. And these polyphenols, they can react with one electron oxidants, as I mentioned before, to actually prevent free radical formations. And this ability of these compounds to have interacted with metal ion has significance in their bioactivity, and polyphenol include phenolic acid, flavonoid, and anthocyanins, which all of these elements, phenolic acid, flavonoid, and anthocyanin, are being studied in my study. So for phenolic compound, so it is actually main class of secondary metabolites in plants that is divided into phenolic acid, gallic acid, salicylic acid, catechol, resorcinol, cresol, hydroquinone, and eugenol. And many studies have shown that actually there's a strong and positive correlation between the phenolic compound contents and antioxidant potential of fruits and vegetables. Whereas we can say that if the phenolic compound is higher, it should be the concentration or the antioxidant activity also higher, because it has been said by Dabrowska. It is actually have a strong a positive correlation between these two elements. So the antioxidant activity of phenolic compound is actually attributed to the capacity of scavenging free radicals, donating hydrogen atoms, electrons, or shielding metal ions. Alright, so anthocyanins is another element that I'm studying, and this element is responsible for the colors. So as you can see, it's contributing to the red color, purple color, and blue color in fruits and vegetables. So few examples are berries, currants, grapes, and some tropical fruits have that have high antioxidant content. So that's why if you can see fruits that have darker color in red, in purple, and in blue, they would significantly have higher anthocyanin content in the fruit of vegetables. So ready to publish blue-colored leafy vegetables, grains, roots, and tubers are the edible vegetables that contain a high level of anthocyanins, depending on the color. So among the anthocyanin pigment, cyanidin triglycoside is a major anthocyanin found in most of the plant, and scientific studies such as cell culture studies, animal models, and human clinical trials have shown that actually anthocyanidins and anthocyanins possess antioxidant and antimicrobial activities that will eventually improve visual and neurological health and protect against various non-communicable diseases. Flavonoid is another element too that I'm studying in my research, and this flavonoid is now has been considered as an indispensable component in a variety of nutraceuticals, pharmaceuticals, medicinals, and cosmetic applications, where this characteristic is being attributed to the antioxidative, anti-inflammatory, anti-mutagenic, and anti-carcinogenic properties, coupled with the capacity to actually modulate cellular enzyme function. And many research on flavonoid have received an added impulse with the discovery of the low cardiovascular mortality rate. So, for the justification of my research, and after I have mentioning all the facts just now, it should be suggesting that the apple peels may possess more bioactivity than the flesh. But interestingly, the interesting part is that we human, we always discard the peel part rather than eating both peel, and it is actually significantly and has been studied that peel has a higher concentration of those elements, but we practice it wrongly, and this practice even being observed higher during the apple manufacturing, whereby we always discard all this peel apple part. Okay, so objective of my study is actually to study the concentration of total phenolic compound, flavonoid content, anthocyanins, and antioxidant in peel and flesh. So basically, I'm going to be just to see whether the peel has higher, the peel is actually have the same concentration, or is differently from the flesh. And the second one is to get to know which part of fruit has a higher concentration of this element. So at the end of this research, I'm going to prove that which one of this part, either peel or either flesh, has a higher concentration of this mentioned elements. And the third objective is to actually decide which cultivars. Since I'm using 5 cultivars, as you can see, Alice, Brenda, Katie, Doreen, and Emma. So I'm going to be coming out with which one of these cultivar is actually best to be consumed regarding to the concentration of this element that I have come out in the result. So for the methodology and materials, actually for the samples and extractions, I am going to be having the flesh or peel obtained from 5 randomly selected apples, where in each trial to minimize variations, and 3 independent extractions were carried out using 10 grams of apple flesh or peel extracted using 100 ml of acid and water, and then the extractions will be made into 5 replicas of samples made from 5 fruits. So the determination of total phenolic content is actually measured using Follin-Ciozel-2 method, and then the total phenol will be expressed as milligram gallic acid equivalent to 100 gram fresh matter. For the determination of flavonoid content, it had been estimated using the modified P dimethylaminocinnamaldehyde or DMACA method, where this total flavonoid content was expressed as mg catechin equivalent to 100 gram fresh matter. For determination of anthocyanin content, it was actually measured using spectrophotometric pH differential protocol that has been reported by Gayasti and Roslet and will be expressed as milligrams cyanidin trifactorside equivalent to 100 gram fresh matter. So the last but not least, the evaluation of antioxidant activity is using the DPPH assay, the modified method of Brand Williams et al. And then it will be calculated by the formula is listed down here. All data were reported as mean standard error of 5 replicates, where all the differences between means will study using two-way analysis of variance, and differences at 5 level percent will be considered as statistically significant. So as you can see from my result, I have conducted the ANOVA two-way ANOVA tables, and I have made it into two different tests whereby I tested on polyphenolic compounds and antioxidant activity differently because polyphenolic compounds and antioxidant activity they come out with different units. So I needed to do both of them differently. So for this ANOVA test, it's actually I want to see for the p-value of the sample. So as the sample here is the peel and the flesh part. So I was actually trying to prove that there is a significant difference between the peel part and the flesh part in terms of polyphenolic compound and antioxidant activity. Whereby I said just now, if the p-value is less than 0.05, it is considered a significant difference. And both results, as you can see in polyphenolic compound, it shows lesser than 0.05, as well as antioxidant activity, it showed lesser. So the first objective is achieved here, whereby I can say that there is a significantly difference between peel part and flesh part, but still I yet don't know, I yet didn't know which part has higher. So it comes to the second test, whereby I so the table uh, I made it into graph from this table. So as you can see, it is significantly higher. It is significantly different from peel and part. Okay, so to test my second objective is that to have seen which part is actually higher. So these are the tables that contains the value of the mean and the standard error mean. Alright, so I've interpreted it into the graph as you can see here. So the trend is higher actually, it is observable actually, that the green color shows higher reading or higher concentrations of rather than the orange one, whereby the green color indicates the peel part. So for the total phenolic content, the peel part has higher and greater concentration of gallic acid. For the total flavonoid that is being measured in mg catechin equivalent to 100 grams also show a different trend to the same trend, whereby you can observe here that the green color has higher reading than the orange part, where it indicates peel part has higher concentration and total flavonoid. The same thing goes here for the anthocyanins, where the green color shows higher reading, where it indicates peel part have higher concentrations of anthocyanins. So for the polyphenolic acid, you can tell that peel part have higher concentrations in rather than in the flesh part. Alright, so let's get to look at another element that I'm actually studying in this research of mine, which is this free radical scavenging activity and percentage. So these are the tables I have interpreted, and I've also interpreted into the graph. So as you can see, there are actually 4 graphs. This is because I have conducted differently in different concentration regarding 0.625, 1.25, 2.5, and 5.0 milligram per milliliter of DPPH. Alright, so across the concentration, you can see actually the blue color has higher concentration or reading to be compared to the flash one, which it indicates that for the DPPH or the free radical scavenging activity in percentage, it also shows a significant difference between the peel and the flash, and it is being proven actually the peel has a higher and greater concentration rather than in the flash. So for the second objective here, we can say that actually the peel part itself has higher concentration and higher reading, which is better than the flash part in terms of the polyphenolic compounds and as well as uh, this free radical scavenging activity in the peel rather than the flash. So, without further ado, let's study this third objective of me to actually determine which cultivar. Since I'm using 5 cultivars, so I'm going to be determining which cultivar is better to be consumed. Uh, so, as you can see from this graph, I actually have eliminated the flash readings because I actually have known that the flash is lower or is the peel part is greater than the flash part. So I just included the peel part for my comparison between cultivars. So as you can see here, the concentration across the concentration from lower to higher, from 0.605 to 5 milligram per milliliter, it shows that the concentration or the reading is higher in the KT cultivar across the concentrations of DPPH assay. So the percentage is higher, is observable in KT cultivar in every concentration that I have tested. So same goes with the polyphenolics in 5 different apple cultivars here. So as you can see, for the total flavonoid, KT has shown significantly higher rate, significantly higher, the highest actually, to be compared to the other cultivars, Alice, Brenda, Doreen, and Emma. And the same has been shown in anthocyanin content and as well as the total content, whereas KT has the higher and greatest reading and concentrations of the polyphenolic compound that is contained the total flavonoid, anthocyanins, and total phenolic content, whereby from these two graphs we can say that across the cultivars, KT has higher reading or higher or the better concentrations, which is good to be consumed. Uh, the reflecting the better concentration or the higher reading of these elements I just mentioned. So for the discussions, the total flavonoid content, total flavonoid, anthocyanin content, and antioxidant activity actually they differ significantly depending on the fruit portions and the variety of apples examined. An apple peel extract had a richer variety of phenolic constituent than apple flesh, as can be seen from the results, and both polyphenolic contents and antioxidant activity values of apples had showed significantly higher in the peel rather than in the flesh. And this study or this result of mine is actually being supported by Derogodi, whereby he come up with a similar trend that he observed in his study when he studied the cultivars in different countries and the total phenolic content of apple parts. Thus, his study that has shown positive health beneficial effects of consuming apple with peels that he reported confirm my work. So for other apple cultivars, Dabrowska also stated the same thing. He said that higher concentration of phenolic compounds and antioxidant activity in the peel than the flesh he documented. Okay, so the finding of this research were actually consistent with a number of previous studies reporting that apples have high antioxidant activity, antioxidant ability, and that the value is actually different between cultivars. And according to Pitkovsak, the explanation of the relatively higher of polyphenolic amount and antioxidant activity could be probably due to the trees of fruit that are actually subjected to numerous stress factors in orchards besides types, diseases, pests, or lack of mineral nutrients that induce phenolic compound accumulations. He actually came up with this explanation when he conducted a comparison of apple cultivars that were susceptible and resistant to fruit scab infections, whereas throughout his experiment, he observed that the uh, the higher the phenolic content is actually observable in the fruit that is resistant to the fruit scab infections. So he came out that total phenolic content in apple fruit resulted in higher total antioxidant activity. Lastly, there is always the difference in the presence and the distribution of this phenolic between the flesh and the apple peel. But tendency apple peel to actually contain significantly higher overall antioxidant activity, and the total phenolic has clearly indicated that peel removal during manufacturing may induce more severe nutrition losses than other cultivars before eating. So for the conclusion, actually, I have proven that it has significant differences of concentration of photophenolic compound, flavonoid content, anthocyanin, and antioxidant activity in the peel and the flesh, where I further confirm and I further prove that actually the peel part of apple clearly has high concentration of these elements. And from this experiment too, I have came up with which cultivar is actually the best to be consumed, and it is proven that KT cultivar is the best and has the highest rating of all factors and considered to be the best to be consumed. So that's all from me. These are my references. Thank you so much.